After studying this module, you shall be able to know the basic structure of amino acids, know different ways of classification, understand physical and chemical properties of amino acids, understand how amino acids behave both like an acid and bases, learn about reactions carried out by amino acids and carboxylic group of amino acids, know about various functions and biological importance of amino acids. Now, amino acids, they play central role both as a building block of proteins and as intermediates in metabolism. 20 different amino acids are commonly found in proteins. The first to be discovered as asparagine in 1806, the last of 20 is found to be threonine, was not identified until 1938. Structure of amino acids. All 20 amino acids, they have carboxylic group and amino group bonded to the same carbon, the alpha carbon, and generally called as alpha amino, alpha carboxylic acids. They differ from each other in the side chain or R groups, which vary in structure, size, electrical charge, and which influence the solubility of amino acids in water. In addition of these 20 acids, there are many less common ones. Some are residues modified after a protein has been synthesized, other are amino acid present in living organism but not as constituents of protein. The additional carbon in an R group of amino acids are commonly designated as beta, delta, epsilon and so forth, proceeding out from alpha carbon as shown. Classification of amino acids based on nature of R group. Aliphatic amino acids, they have aliphatic chain as their R group and 5 amino acids are included in this as glycine, alanine, valine, leucine and isoleucine. Glycine provides a minimum of steric hindrance to protein, to rotation and to placement of adjacent group. Valine, isoleucine and leucine, the bulky groups of this participates in hydrophobic interaction of proteins and they are found present in the interior of the protein. Hydroxy amino acids, they have hydroxyl group in their R group and the examples are serine and threonine. Serine and threonine, OH group is weakly acidic which is having a pK of 13.6. It can form esters with the phosphoric acid or organic acid. It is a site of attachment of a sugar rings in glycoprotein. Hydroxyl group present in serine are found at the active center of some enzymes. Sulfur containing amino acids, they contain sulfur group in their R group. Examples are cysteine, methionine and cysteine. 2-cysteine SH group can be oxidatively joined to form a disulfide bridge in a double headed amino acid cysteine. Acidic amino acids, they are dicarboxylic acid and containing an additional carboxylic group in their side chain and also exist in their corresponding amides. Examples are aspartic acid, asparagine, glutamic acid and glutamine. Carboxylic group present in the side chain of aspartic acid and glutamic acids are dissociated at neutral pH. It is having a pK of 4.3 to 4.7 and provide anionic group on surface at protein. Basic amino acids, they contain additional amino group in the heterocyclic ring is also present. The examples are arginine, lysine and histidine. Imidazole group in histidine chain are a part of active site of many enzymes. Like other basic group in protein, they bind to the metal ion. The flexible side arm with a potentially reactive amino group at end is present in lysine, which is having a pK of 10.5 means that lysine side chains are protonated in neutral solution. The guanidinium group in side chain of arginine has a pK of 12 and remains protonated under most circumstances. Aromatic amino acids, they have aromatic R groups and the examples are phenylalanine, tyrosine, tryptophan. Tyrosine, tryptophan and phenylalanine can form hydrophobic bonds and especially effective in bonding to other flat molecules. Hydroxyl group present in tyrosine has a pK of 9.5 to 10.9 and act as hydrogen donor in hydrogen bonds. Amino acids. The amino group is present rather than the amino group, and the example is proline. Proline has got secondary amino group which makes a relatively rigid conformation, and the presence of proline strongly influences the folding of protein chains. Based on polarity of side chains, amino acids are classified on the basis of polarity of the side chain as nonpolar side chain, as a polar side chain, the polar side chain with a positive side polar side chain, and with a negative polar side chain. The nonpolar amino acids, they contain polar, aliphatic or aromatic side chain. There are 8 examples. Nonpolar side chains are usually hydrophobic and in deep buried into the interior of protein molecule. Polar amino acids, they have hydrophilic side chains which form hydrogen bond with water. They have groups like hydroxyl, amide, thiol, phenol, and these group may be charged or uncharged. Based on nutritional requirement, according to the nutritional requirement, amino acids are classified into essential, semi-essential and non-essential. Essential amino acids are those which are not synthesized in our body. They are required to be taken in diet because they have essential function in our body. Semi-essential. The semi-essential amino acids are inadequately synthesized in our body. They are able to partially meet daily requirements. However, their deficiency occurs in condition of increased demand such as during growth, pregnancy and lactation. So, our body partially meet their demand. These are arginine and histidine. Non-essential amino acids, these non-essential amino acids are adequately synthesized in our body. They need not to be taken in the diet, hence called non-essential amino acid. Metabolic classification for the amino acids. On the basis of their catabolic end products, the 20 standard amino acids are divided in 3 groups: glucogenic amino acids, those which can be converted into glucose. 14 out of 20 standard amino acids are glucogenic amino acids. Ketogenic amino acids, those which can be converted to ketone bodies, example acetone, beta hydroxybutyrate and acetoacetate. Both glucogenic and ketogenic, those which can be converted to both glucose and ketone bodies. Some uncommon amino acids. These modified amino acids are those which are modified after they are incorporated into the proteins, as proline and lysine undergo hydroxylation to form hydroxyproline and hydroxylysine. These are essential for the formation of natural collagen molecule. Methylated and acetylated amino acids are present in histone proteins and ribosomal protein. 6-methyl-lysine is a constituent of myosin, a contractile protein of muscle, and gamma carboxyglutamate found in blood clotting protein. More complex desmosine made up of 4 lysine residues found in fibrous protein elastin. Modified amino acids. These modified protein are those which are modified after they are incorporated into the proteins. Example, hydroxyproline and hydroxylysine. Proline and lysine, they undergo hydroxylation to become hydroxyproline and hydroxylysine. These are essentials for the formation of mature collagen molecule. Methylated and acetylated amino acids are present in histone proteins and ribosomal protein. Some non-protein amino acids. Some 300 amino acids have been found in cell. They have variety of functions but are not constituent of protein. Ornithine and citrulline are the special examples. They are intermediates in biosynthesis of arginine and constituent of urea cycle. Non-amino acids. Some amino acids are typically amino acids but are amino acids because they have NH2 group attached to carbon atom rather than alpha carbon atom, and the gamma amino butyric acid or the beta alanine are the examples of this. Selenocysteine is a special class, is introduced during protein synthesis rather than created through post synthetic modification. It contains selenium rather than sulfur of cysteine. Selenocysteine is a constituent of few known proteins and recognized as 21st amino acid. We have symbolic representation of amino acids. Abbreviations and symbols are commonly used for the occurring amino acid. Each amino acid has a name associated with 3-letter abbreviations and a 1-letter symbol. The 1-letter codes are determined by the following rules. That is a unique first letter. If only one amino acid begins with a particular letter, then the letter is used as a symbol. For example, I is for isoleucine. Most commonly occurring amino acids have priority. If more than one amino acid begins with a particular letter, the most common of this amino acid receive this letter as a symbol. For example, glycine is more common than glutamate, so G goes with glycine. Similar sounding amino acids. Few 1-letter symbols like the amino acids they represent, for example, F is for phenylalanine and W is for tryptophan. Letter close to initial letter. For the remaining amino acid, a 1-letter symbol is that as close to alphabet as possible to the initial of that amino acid. Further, B is assigned as asparagine, signifying either aspartic acid or asparagine. Z is assigned as GLX, signifying either glutamic acid or glutamine, and X is assigned to an unidentified amino acid. Now, the properties of amino acid. The amino acids show optical property. All amino acids except glycine have at least one asymmetric carbon atom which is attached to 4 different groups. Hence, all amino acids are optically active except glycine, which is optically inactive. Aromatic amino acids, they show absorbance at 280 nanometer, and tryptophan has got a maximum absorbance. D and L amino acid. Nearly all biological components with the chiral centers occurs naturally in one stereoisomeric form or the other. The amino acid residues in protein molecules are exclusively L-amide stereoisomer. D amino acids have been found only in few, generally small peptides, including some peptides or bacterial cell wall, and in certain peptide antibiotics. Example, actinomycin. Human body, however, neither synthesizes nor uses D amino acid. Amide group is not acidic but polar and participates in hydrogen bond. 2 cysteine groups can be oxidatively joined to disulfide bridge in double amino acid. Acid based behavior of amino acids. Amino acids in aqueous solutions contain weakly acidic carboxylic groups and weakly basic amino groups. In addition to each of acidic and basic groups, amino acids contain an ionizable R group in a side chain in some of the amino acids. In free amino acids, the carboxylic group and the amino group of the general structure are charged at neutral pH. The carboxylate portion negatively and amino group positively. Amino acids without charge group of their side chain exist in neutral solution as Zwitter ion with no net charge. A Zwitter ion has equal positive and negative charge in solution and it is electrically neutral. Substances having this dual nature are called amphoteric and called ampholides. A simple monoamino monocarboxylic acid such as glycine is diprotic. When fully protonated at acidic pH, it has 2 groups, the COH and NH3 positive group that gives proton. Ionization of amino acid. The cationic form in the low pH form and the titration of cationic species with base yield the Zwitter ion, and finally the anionic form. The titration curve of an amino acid and pI. The plot of pH against the amount of NaOH added to given acid is called a titration curve. When amino acid is titrated, its titration curves indicates the reaction of each functional group with hydrogen ion. The titration curve or the plot has 2 distant stages corresponding to deprotonation of 2 different groups on glycine. First stage, at very low pH, the predominant ionic species of glycine is fully protonated form. At the midpoint, in the first stage of our titration, in which COH group of glycine loses its proton and equimolar concentration of proton donor and proton expector species they are present. At the midpoint of any titration, a point of inflection is reached where the pH is equal to pKa of protonated group being titrated. For glycine, the pH at midpoint is 2.34 as COH group has a pK of T through 2.34. pK is the measure of a tendency of a group to donate proton and denoted as minus log of Ka, and tendency to donate a proton decreases 10 times as pK increases by 1 unit. At the titration curve proceed, reached at pH 5.97, then is another point of inflection at which the removal of first proton is essentially complete and removal of second has just begun. At this pH, glycine is present as a dipolar ion and point is called Pi. The second stage of titration corresponds to a removal of proton from the amino group of glycine. The pH at the midpoint of this stage is 9.6, equal to the pK of amino group. The titration is essentially complete at pH 12, at which the predominant form of glycine is negatively charged. From the titration curve of glycine, we can derive several important information. 1. It gives quantitative measure of pK of each of two ionizing group, that is 2.34 for carboxylic and 9.6 for amino group. It also tells that amino acid has got two buffering zone. One of this is relatively flat portion of curve extended for approximate pH unit on either side of the pK 2.34, indicating that the glycine is a good buffer near this pH. The other buffering zone is around pH 9.6. It also gives the relationship between an electric charge and pH of the solution. At pH 5.97, the point of inflection between the two stages in a titration curve, the glycine is mainly present as a dipolar ion, fully ionized but with no electrical charge. The characteristic pH at which the net charge becomes equal to 0 is called the isoelectric point or isoelectric pH, designated as Pi. Isoelectric point is simply the arithmetic mean of two pK values. Here the Pi is half of pK1 plus pK2, which is equal to half of 2.34, which is the pK for carboxylic, plus 9.6, which is the pK for amine group. In alanine or many other monoamino carboxylic acids, the carboxylic groups and amino group are two titratable groups. The titratable group of each amino acid has got the characteristic pK value and it tells about the strength of an acid. The pK value of amino groups are much higher with values ranging from 9 to 10.5. When the titration is started, amino acid, diprotic acid, base NOH is added. The carboxylic group loses its proton. First, it has a low pK value in comparison to the amino group and hence stronger acid and become negatively charged carboxylate ion, and the pH of the solution increases. As the pH increases still further, the addition of more NaOH is protonated amino group weak acid loses proton and alanine has a negative charge of -1. Amino acids, they differ in their acid base property. The all amino acids with a single amino and single carboxylic group that does not ionize have a titration curve resembling that of glycine. The amino acid with an ionizable R group have more complex titration curves with 3 stages corresponding to 3 possible ionization, thus have 3 pK values. The titration curve of glutamic acid will give 3 flat zone. Importance of Pi. Amino acids in their zwitterionic form have least solubility and are precipitated at their Pi. This property is used to separate an amino acid mixture from the mixture of several amino acids. This can be done by adjusting the pH of a solution of a mixture of amino acid equal to the pi of amino acid which is to be separated. At pi, an amino acid can act as a buffer because a Zwitter ion can donate a proton as well as accept a proton. Zwitter ions, they do not move in electrical field because there is no net charge. How to determine isoelectric point of an amino acid? Pi of a simple monoamino monocarboxylic acid is usually midway between the two pK values, pK1 that is for the carboxylic and pK2 that is for the amino group as discussed. Pi of an amino acid is more dissociable group other than alpha amino and alpha carboxylic can be determined in a similar way. For example, aspartic acid has got an other COH group. In all, it has 3 dissociable group with different pK values. For alpha amino, pK is 9.82, for alpha carboxylic pK is 2.09 and for gamma carboxylic it is pK is 3.86. Dissociation of aspartic acid on titration with NaOH gives following reaction, and the Pi is taken as the mean of 2 carboxylic group pK1, pK2 divided by 2. Now looking at the chemical reaction of amino acid. All amino acids undergo several chemical reactions involving different group. First we see the reactions of amino group. The amino group can undergo transamination reaction. The alpha amino group of amino acid is released to form a keto acid. A different keto acid accepts the amino group to form a new amino acid. Alpha ketoglutarate and pyruvate are the main keto acids which accept the amino acid to form a glutamate or alanine. Deamination, the removal of alpha amino group from an alpha amino acid is called deamination. Usually it is a oxidative deamination in which alpha amino group is removed from an amino acid with its simultaneous oxidation and leads to the formation of keto acid with the liberation of ammonia. Most amino acids can undergo transamination, but very little oxidative deamination. The glutamate is the only amino acid which undergoes both. The combined transamination and the oxidative deamination leads to removal of ammonia and the transport of liver for urea formation. Reaction with ninhydrin, the amino group of free amino acids or N-terminal of proteins react with ninhydrin reagent to give a purple color complex. This is a very sensitive test and it is used to detect the presence of free amino group. Reaction of carboxylic group, decarboxylation. In decarboxylation, the COH is removed as CO2 to form amines, and this reaction is catalyzed by decarboxylase enzyme. Several important amines are produced from amino acids. Decarboxylation, like for example, histamine produced from histidine, serotonin is produced from tryptophan. Importance of amino acids. First, the function of proteins, formation of proteins. Amino acids are joined to each other by the peptide bond to form a protein, formation of glucose. Glucogenic amino acids are converted to glucose in body. Enzyme activity, the thiol group of cysteine has an important role in certain enzyme activity. Transport and storage form of ammonia. Amino acids, glutamine plays an important role in the transport and storage of amino nitrogen in the form of ammonia. They act as buffer. Both free amino acids and some amino acid present in protein can potentially act as a buffer. Histidine can serve as a best buffer at the physiological pH because it has got the pK of 6. Detoxification reaction, glycine, cysteine and methionine are involved in the detoxification reaction. And formation of biological important compounds, specific amino acids can give rise to specific biological important compounds like tyrosine can give rise to some hormone, tryptophan can give rise to vitamin niacin, glycine and cysteine they can make bile salts, and beta alanine can give coenzyme A. Summary: each amino acid has got a alpha carboxylic group and amino group except for proline which is got the imino group. Each amino acid also contains one of the 20 distinctive side chain attached to alpha carbons. The amino acids can be classified on the basis of their R groups, nutritional requirement, the polarity, the metabolic products of amino acid. The alpha carbon of amino acid except glycine is asymmetric and can exist in stereoisomeric form DLL only, and only L amino acids are found in protein. The amino acids can behave both as base and hence can act as buffer. Histidine serves as the best buffer near the physiological pH. Amino acids have got various function other than proteins, and amino acids can give rise to various biologically important compounds. Amino acids, they can show various titration curve which is useful in predicting pK values and finding out Pi values, and the amino acids at their Pi values show net zero charge and called Zwitter ions.